So Genprex, our mission is to reprogram the course of cancer. We're a publicly traded company, and so there are some risks to the development of this technology, and I encourage you to look at the SEC to look at the risk factors. So we are a 21st century gene therapy for cancer. Gene therapy, as you know, during this conference has been around for decades now, but finally the technology has caught up, and we're now able to use this technology for the purpose of treating hard-to-treat cancers, and particularly our focus is in solid tumors. So we have developed a cutting-edge, first-in-class immunogene therapy. Immunogene because it is a gene therapy, but it has features of immunotherapy as well. And we encapsulate this API inside a proprietary lipid nanoparticle that is non-viral. We believe that there are significant market gaps as well as unmet medical needs, especially in patients who had advanced lung cancer. And so our first indication is non-small cell lung cancer. It's the most common cancer and the most deadly. You can see the numbers here. Unfortunately, the 5-year survival for stage 4 non-small cell lung cancer is still abysmal, around 1%. We are looking at patients who don't necessarily have the right molecular profile for some of the more modern treatments, such as immunotherapies or some of the EGF receptor inhibitors. And so our target population are those patients who have failed prior therapies or don't have, for example, an EGF activating mutation to allow them to be treated. We have a platform that consists of multiple tumor suppressor genes, numbering 10, that we encapsulate and use in synergy with some modern therapies, such as EGF receptor inhibitors, immunotherapies, et cetera, et cetera. And the goal will be in the future to use these synergies to treat multiple types of cancers, and solid tumors in particular. So how the technology works. The tumor suppressor itself, once we are able to deliver it inside the cell, acts as a pan-kinase inhibitor. So as you can see, it acts downstream of various growth factor receptors, such as EGF receptor, and inhibits proliferation. It also stimulates apoptosis. And what we've found, and this is the immunomodulatory aspect of the drug, is that it stimulates the cell in a way to make it, the environment more immunogenic. And the key, of course, is how do you deliver the drug into the cancer cell? And so we have developed and optimized a non-viral lipid nanoparticle. It is a cationic, so positively charged, small vesicle that is able to contain the TUS2 gene. TUS2 is our API. And this nanovesicle is able to home into cancer tissue preferentially and thus kill those cells through various mechanisms, including apoptosis. I won't go into the details of mechanisms, but this technology came out of MD Anderson and is well characterized in terms of various pathways in which it is active. So where are we in our clinical development? So you can think of us as having two major programs. One is our drug, Genprex, in combination with erlotinib, which is a first-generation EGF receptor inhibitor. And this program we've taken to an early phase 2, and I have some data to show you. And the next program that we're very excited to present and to start in the coming year will be Genprex plus an anti-PD-1. We are hoping that with the interim data that we have in our Genprex plus erlotinib program, that we can get into a fast track or accelerated approval designation. So this is a design of our phase 2 trial. It was actually a combination trial of Genprex plus Tarceva. It was designed as a dose escalation of both drugs, and you could see at the bottom the dose levels. We had 3 different dose levels, which were increments of erlotinib or increments of our drug Genprex. The majority of the patients were EGFR negative, so typically these patients don't benefit from an EGF receptor inhibitor. Even that one patient of the top who was EGFR positive but with a rare mutation, you can see that patient had a prior line of therapy using chemotherapy and not a targeted therapy. So we exposed all these patients to our combination drug of Genprex plus erlotinib, and these patients who were progressing on prior therapy, we were able to buy them extra weeks. They had stable disease for 10 plus weeks, and in the phase 2 portion of the trial, we even had one patient who had a complete response and out for 30 plus weeks. And so our strategic objectives in the coming year will be to continue this trial at MD Anderson and other sites, to pursue strategic partnerships, looking at our science and synergies, and to investigate the use of Genprex in other indications, as well as develop our technology a little bit further. So I'll stop here. Give us about 8-9 minutes or so. Thank you.
Great. So, Julien, my first question is for your Genprex constructs. How do they achieve, just diving deeper into the design, how do they achieve selectivity and avoid any off-target effects?
Yeah. So the nanoparticle itself takes advantage of a phenomenon called enhanced permeability and retention. So it's been well known that certain nanoparticles, lipid nanoparticles specifically, who have certain physical chemical properties, can sneak through immature vessels or immature vasculature that usually forms around cancer cells or cancers or large cancer masses. And so we have found through pre- and post-treatment biopsies in our phase 1 trial that there's a 10 to 25-fold uptake of the drug preferentially in cancer tissue compared to normal tissue. So even though this is a passive mechanism, we've seen some tropism towards cancers, especially vascular cancers. And I think this is an argument for us in the future to explore other types of cancers who also happen to be quite vascular.
Great. And then, with your delivery approach, talk about the advantages that might provide you in terms of manufacturing.
Yeah, so speaking to many of my peers who sometimes don't have their own manufacturing infrastructure, as you know, making a viral vector sometimes puts you out a year and a half. We have put so much resources, so many resources in trying to thread that needle and find all the CMOs that can partner with us for the manufacturing of this complex drug. And it turns out that the majority of the vendors, for example, that we speak with can get us into the suite within 8 to 12 weeks compared to if we were to use a viral vector. So that's been one of the advantage in terms of manufacturing. And as you heard during one of the panels this morning, manufacturing is really one of our key differentiators in that we have a well-characterized process and method that we are going to optimize and scale up out of MD Anderson into and hopefully get into the clinic soon.
And in the evolution of advancing LNPs, a lot's been learned in how to avoid toxicity. So, what of that learning have you guys been able to take advantage of?
So we have given this drug either in phase 1 or the current phase 1/2 trial to almost 60 subjects so far. And out of this small sample of patients, we have not seen a significant degree of adverse events. What we've seen have been either adverse events related to Tarceva, usually rashes, and we've seen a little bit of lymphopenia, and hypophosphatemia has been kind of one of the adverse events that we've seen. So the technology itself has been I mean, the tolerability of the drug has been well characterized for us, and yeah, we feel that this technology is going to be a solid platform for us to start putting various tumor suppressor and explore other indications.
And just expanding on the phase one, what other signals of efficacy have you seen in the study?
Yeah. So even in a phase one monotherapy trial that we concluded in 2012, in that trial we had 31 patients enrolled, and I believe 27 who had 2 or more doses, so as a dose escalation scheme. And out of these patients, 5 of them had stable disease or better, and 3 had a significant response, a durable response. We had 1 patient who survived many, many years beyond this, so she was a little bit of an outlier. But I think it was this element that really allowed us to understand the MTD for this drug and how well tolerated it was. And even in the current trial that we're doing, the phase 1 expanded to phase 2, we've remarked that we were able to accomplish the same MTD in this particular trial. So far, it looks like we are on target in terms of finding the right dose for patients and not seeing some balance of efficacy or signal of efficacy and good tolerability.
And what else are you learning from this study, either in terms of safety or efficacy, that will inform how you design or go after future studies, especially if you want to see how your product works in combination with other targeted therapies for the lung?
So Dr. Roth, who's our scientific founder, is right now at MD Anderson, is right now focusing on the synergies of our agent Genprex in combination with immunotherapy, and so he's explored this drug in humanized mouse models, in combining with the Pembro or Opdivo, and has found that we're seeing a signal of survival in mice, in these humanized PDX models, and that there's clearly an immunogenic effect. There's an increase of natural killer cells and CD8 positive T cells. So, we are really encouraged and excited about these preclinical findings, because we believe that being able to synergize with what is going to be the backbone of cancer therapy in the future, i.e. checkpoint inhibitors, will allow Genprex to really be well positioned. If you look around, I mean, if you look at the current guidelines for the treatment of non-small cell lung cancer, there's really 2 major arms, right? 2 pathways. If you have a driver mutation, and most often it's an EGF receptor mutation, then you get treated with an EGF receptor inhibitor. And if you don't have an if you don't have a driver mutation, you get treated with immunotherapy plus minus chemotherapy. And so this is one unique drug that actually works on both arms, right? Because we could combine our drug with both a targeted therapy and a checkpoint inhibitor. So we're quite excited about this position.
So finally, in terms of timing, what would be the next data release that investors should be looking forward to?
Yeah, so I can't give specifics on that, but it'll be imminent that we can restart the trial at MD Anderson. We had to put on hold voluntarily for some time in order to scale up manufacturing and other functions. So we will resume recruiting at MD Anderson, and things seem to be going very well from a manufacturing standpoint as well in our scale-up. And so as soon as we feel that we have sufficient drug products, we will expand to other sites. And we've already talked to many PIs, and there's a general sense of excitement about this agent, because many of these oncologists are frustrated in that they can't find options for these patients who have become resistant to immunotherapies or targeted therapies. So no data readout in the near future, but we will be resuming trials very soon.
Okay, well, very good. Thank you so much.
Thanks, Kay.